Welcome to part 17 of this video on the immune system. Okay, so we're currently talking about the adaptive immune response, and the point that we've got up to now is that our T cell has received signal 3, and signal 3 is the final signal in T cell activation, and indeed it's actually going to deliver the signal which will activate the T cell. So what I now want to discuss is what is this T cell going to actually do, and for this bit that I'm about to discuss, the pathway will still be the same for both the cell-mediated adaptive immune response and also the humoral adaptive immune response. So I'm not going to distinguish between the two yet. We will have to very soon, but for now the pathway remains the same whether we're talking about a CD8 positive T cell and therefore the cell-mediated adaptive immune response, or a CD4 positive T cell and therefore the humoral adaptive immune response. Okay, so we'll go over onto a new sheet of paper and then let's discuss what this T cell that has received signal 3 is actually going to do. So let's just draw it here. So here's our T cell and it's just received signal 3. What's going to happen now? Well, now it's going to start dividing and dividing and dividing and dividing. So it's going to begin a process of proliferation and it's going to produce loads of T cells that are all identical to it. And the crucial part of them all being identical to it is that they're all going to have the same design of T cell receptor. So they're all going to produce identical T cell receptors. So I'll draw a little population here. In reality, you'd be producing far more than just 4 copies of this T cell. But for the basis of this picture, 4 will do. Okay, so these are the progeny of this T cell and they are all genetically identical to it, and they all have the rearranged alpha chain locus and the rearranged beta chain locus, and therefore they're all going to produce identical T cell receptors to the original one. So what have we done here? We've expanded this clone of T cells. Remember, a T cell clone is the name for the population of T cells that all have an identical T cell receptor. So we have effectively expanded this T cell receptor clone here. Okay, and for that reason, this process is known as clonal expansion. So after signal 3, what will occur is clonal expansion, where the signal 3 activated T cell will divide and divide and divide and produce loads of progeny which all have the identical T cell receptor. So we've got clonal expansion, and of course this will be occurring in the lymph node that the T cell was present in when it was activated by the antigen presenting cell. So you'll produce this large clone of identical T cells in a little area in one of your lymph nodes that is draining the affected or infected area. Okay, right. So what's now going to happen? What are all of these copies of this T cell that we've produced here actually going to do? Well, they're now going to start differentiating, and most of them are going to become what are known as effector T cells. Some of them are going to become memory T cells, and what I want to discuss is what the memory T cells are going to do, because that's the bit that's common for both CD4 positive and CD8 positive T cells. Then to discuss what the effector T cells are going to do, we're going to have to split the story into the cell-mediated story and the humoral story. So this is where the two stories are going to diverge. Okay, but one last step that I can discuss together is the differentiation step. So these cells are now going to differentiate from being mature naive T cells, which aren't actually doing anything at the moment. They're not actually activated to do anything, they're just sitting and waiting, and they're now going to differentiate into T cells that are capable of doing something known as the effector T cells, or they're going to differentiate into a type of T cell known as a memory T cell, and far more are going to differentiate into effector T cells than memory T cells. So I'll have one out of the 4 differentiating into a memory T cell, and the other 3 can differentiate into effector T cells. Okay, so once again, here are my 4 cells, and I'll have the effector T cells, I think, in red. So I'll circle them in red here, and we'll discuss what these are going to do in just a moment. As I say, we'll have to distinguish between the cell-mediated adaptive immune response and the humoral adaptive immune response to discuss the effector T cells. So these in red these are now the effector T cells of either the CD4 positive type or the CD8 positive type. And then the final cell that we've got up there, the other thing that the naive, mature T cells can differentiate into is what are known as memory T cells. So this in green here, this is representing what's called a memory T cell. Okay, so what is the job of memory T cells? Well, their job is very similar to the mature naive T cells. The major difference is that they have a much longer life than mature naive T cells. So the idea here is that when you expand the clone here, the T cell clone here, you don't want all of the T cells from that T cell clone just becoming effector T cells. Can you see why that would be a problem? If they all became effector T cells have a very short lifetime. They're going to be used in the adaptive immune response to hopefully eradicate the pathogen, but very quickly after we've completed the eradication of the pathogen, they're then going to be cleared. Now, if the entire clone of T cells with this T cell receptor have become effector T cells, that means that after we've done that adaptive immune response and cleared the pathogen, we'd lose that entire clone of T cells because they'd all be in the effector T cell form and they'd all now have a very short lifespan and they'd be cleared from the bloodstream. And if we're an adult, we're not going to be able to regenerate that T cell clone because we've lost our thymus gland. So we do not want the entire clone of T cells with this specific T cell receptor to now become effector T cells. So some of them remain as memory T cells, and these perform a very similar function to the naive mature T cells, i.e. they're going to circulate between the circulatory system and the lymphatic system. They're not going to go to the site of infection and attack the pathogen. Instead, they are going to wait around and see if we get infected a second time by the pathogen. They are going to be responsible for what is known as the secondary adaptive immune response. Okay, so they have a much longer lifetime than mature naive T cells. They have a lifespan that can last the entire lifetime of the human. So memory T cells can stay with you for your entire life. And they're responsible for what's known as the secondary adaptive immune response. So what we are talking about is the primary adaptive response. And the reason it's called the primary adaptive immune response is we are assuming that this is the first time you have ever been infected by this pathogen. So what's having to happen is you're getting the antigen fragments presented on on the surface of antigen presenting cells on MHC complexes, and these antigen presenting cells are going to mature naive T cells that have never actually seen their antigen fragment before, and then it's activating them. That takes quite a long time to actually get going. You see, the reason is you have a very small number of these mature naive T cells which have a T cell receptor that can actually be activated by that antigen fragment. So we can say the T cell clone directed against that antigen fragment is very small initially. So it takes quite a while for the antigen presenting cell to actually come into contact with the appropriate T cell, i.e. one of the T cells in this T cell clone. Because after all, it might just be one cell in that makes up that entire T cell clone. It might be a few more than that, but still it's difficult for the antigen presenting cell to actually find that relevant T cell that it needs to find. So that's why the primary adaptive immune response, so the primary response takes so long. So the primary adaptive immune response takes quite long to initiate. When instead you present the pathogen the second time around, so if the pathogen infects you a second time around, the adaptive immune response to that pathogen is initiated much more quickly, and this is now called a secondary adaptive immune response. And the reason it's initiated so quickly is because these memory T cells were produced as part of the primary adaptive immune response, and you produce quite a lot of these, so the size of the T cell clone that is specific against the antigen fragment from the pathogen is now much bigger. So you have far more of these T cells that have a T cell receptor directed against this antigen fragment. So this time it will take the antigen presenting cell much less time to actually find one of these relevant T cells. So I'll stress that the memory T cells, they serve exactly the same function as the naive T cells, except that they live much longer. And you'll have them in a much larger population, you'll have a much greater population of them, and this is the basis for why the secondary adaptive immune response is so much quicker than the primary adaptive immune response, because it takes much less time for the antigen presenting cell to find one of the members of that T cell clone, because it is now a much bigger population of cells than it was at the start of the primary adaptive immune response. So I hope that makes sense. So you produce some of these memory T cells so that if you become reinfected by the pathogen, you have some T cells that are ready to be activated by an antigen presenting cell again. So these are effectively replacing the naive, mature T cells as part of this T cell clone against that specific antigen fragment. Okay, so this will occur for both CD4 positive and CD8 positive T cells. So if we're talking about the humoral or the cell-mediated adaptive immune response, it's exactly the same. You'll get some memory T cells and some effector T cells produced. A lot of effector T cells, but it's very important that you get those memory T cells produced to replace the naive mature T cells and to stick around and be there if we ever get infected by the pathogen again and need to relaunch this same adaptive immune response. And that's why the secondary adaptive immune response is so much quicker than the primary adaptive immune response. Right, so what I now want to discuss is what the effector T cells are going to do, and this is the point where we need to diverge. We need to split between the cell-mediated adaptive immune response to intracellular pathogens and the humoral adaptive immune response to extracellular pathogens, because it now is completely different. So the way I'm going to do it is I'm going to start off with talking about the intracellular response, the cell-mediated adaptive immune response. And the reason I'm going to do that is because it's a much shorter discussion than the humoral adaptive immune response. So I think it's best to do the cell-mediated adaptive immune response first, because it's a nice short discussion and we should be able to finish it in this video. And then we'll go on to the much more hefty topic of the humoral adaptive immune response. And that, I'm afraid, involves talking about B cells, which are, again, incredibly complicated and require a huge, great discussion in their own right. So that's why we're doing cell-mediated adaptive immune response first. So we're now going to look at the cell-mediated adaptive immune response. So let me just make sure that everyone is completely clear with what this is going to be an immune response against. We're going to have some intracellular pathogen. So it's going to be inside your own cells, and then remember what will happen is the antigen presenting cells, they will phagocytose one of your own cells that is infected with the pathogen. Now I'm just trying to find the pictures that I had of this. Here we go. So here we go. So this is a reminder of antigen presentation. So here is our infected host cell here. This is our antigen presenting cell, and it will phagocytose the infected host cell, and it will break the infected host cell down, and by a mechanism that isn't well understood, some of the antigens from the intracellular pathogen will be moved into the cytoplasm undestroyed, so intact. And in the cytoplasm they will then be broken down by the proteasome, which is the machinery inside the cytoplasm of cells for breaking down proteins. So it will be chopped up into little fragments. The fragments will then go into the rough endoplasmic reticulum via the transporter associated with antigen presentation, the TAP protein here, and then they'll be loaded onto MHC class 1 proteins. Remember, MHC class 1 likes to have peptide fragments between 8 and 10 amino acids. And these MHC class 1 proteins will then be transported to the cell membrane through the classical secretory pathway which we went over here. Okay, so the antigen-presenting cells will put the antigen fragments on MHC class 1, they'll then go through the lymphatic system to lymph nodes to activate T cells. And the type of T cell they will be activating are CD8 positive T cells. They will not do anything to CD4 positive T cells. CD4 positive T cells have a T cell receptor which recognizes peptide fragments bound to MHC class 2, and MHC class 1 and MHC class 2 are quite different. So you won't get a T cell receptor mistaking one for the other. Okay, so it's only going to be able to activate CD8 positive T cells, which are also known as the cytotoxic T cells. So we've now had this activation process occur. So the antigen presenting cell was successful. It found a T cell which had the appropriate T cell receptor to be activated by the MHC class 1 with one of these specific antigen fragments in its peptide binding groove, and that cytotoxic T cell has now expanded its T cell clone, so it's proliferated, produced a whole clone of those identical T cells, and now they've started to differentiate. Some of them will become memory cytotoxic T cells, and others will become effector cytotoxic T cells. And it is our job to now understand what these effector cytotoxic T cells are going to do. Okay, so here we go. So let's say this is my effector cytotoxic T cell, or my effector CD8 positive T cell. So I'll just put T subscript C for cytotoxic T cell. Okay, so what are these effector cytotoxic T cells now going to do? Well, they're going to work their way into the bloodstream, and we've discussed how T cells circulate between the lymphatic system and the bloodstream. So they'll get into the bloodstream, and then once they're in the bloodstream, they will circulate around the bloodstream, and some of them will end up going into the microvasculature in the infected area. So let me just draw this here. So let's say that this is the tissue that is actually infected, and just so that I've got something to draw, let's say it is the skin. So here's the epidermis and here's the dermis. So let's say some of these effector cytotoxic T cells that we produce, and I want to stress you will produce absolutely loads of these things. So some of the effector cytotoxic T cells will just happen to go through the microvasculature at the affected area, and then what will happen is, because the microvasculature is currently undergoing the inflammatory response, they'll be recruited into the tissue fluid at that area. And in fact, their mechanism of being recruited is identical to the mechanism by which monocytes were recruited from the bloodstream into the tissue fluid. So they will have sialyl Lewis X on their surface, and I'll just write this down here. They'll have sialyl Lewis X, or SLEX, on their surface, which will then interact with either P-selectin or E-selectin on the surface of the endothelial cells. And remember, that will cause the slowing down of the effector cytotoxic T cell. That'll cause the effector cytotoxic T cell to start rolling along the endothelial surface, because these interactions are very weak, so they can continuously form and break. Okay, that will slow our cytotoxic T cell down, and that will then allow tight adhesions to form. Now, what are the tight adhesions that need to form? Well, on the surface of the effector cytotoxic T cell, just like on the surface of the monocyte, you will have VLA4, and this will bind to the vascular cell adhesion molecule 1, VCAM1, which is on the surface of the activated endothelial cells. Okay, so that's how tight adhesions will form between our effector cytotoxic T cell and the endothelial cells, and then the effector cytotoxic T cell will diapodese across the endothelial layer into the tissue fluid. So this is occurring across the capillaries and the post-capillary venules, and that's the way in which the effector cytotoxic T cell will come into the tissue fluid. And then of course it will migrate to the affected area, just following the gradients of the inflammatory mediators. So let's say that we now have our effector cytotoxic T cell in this area then. So I'll just colour him in red again. So what is it now going to do? What is he actually going to do once he's here? Well, I gave you the answer long ago as to what the cell-mediated adaptive immune response was all about. The power of these effector cytotoxic T cells is that they have the ability to kill infected host cells. That's what their job is going to be. It's going to be to go round, find infected host cells, and tell them to commit apoptosis, to commit suicide. But there's a big question here. How do these cells know which of the human cells that they come into contact are infected with the pathogen and which are not? Because we don't just want this cell to kill any human cell that it comes into contact. That wouldn't be good. We just want it to kill those ones that are infected with the intracellular pathogens. So the big question that we now need to explore is how on earth does it know which host cells are infected with the intracellular pathogen? Okay, so I now need to tell you something that I have hidden from you for a long time now. And this is the fact that antigen-presenting cells are not the only type of cell which can actually participate in antigen presentation. In fact, almost all nucleated cells in the human body are participating in antigen presentation all the time with MHC class 1. They don't use MHC class 2, only the antigen presenting cells, those special cells that we've looked at which are capable of activating the adaptive immune response, so the macrophages and the dendritic cells, only they can use MHC class 2, but nearly all nucleated cells are capable of using MHC class 1, and indeed they do use MHC class 1 all the time. Okay, so let me explain this now. So, let me now take a normal cell. So I'm just taking a normal cell at the moment, not an infected cell at all. All normal cells are doing antigen presentation all the time. Right, so here we have our normal cell, and I'm going to draw its nucleus here with the rough endoplasmic reticulum again, like so. So, nearly all nucleated cells in the body are continually participating in a process of antigen presentation all the time. However, they are not capable of activating the adaptive immune response in the way that the antigen presenting cells are. So antigen presenting cells are the cells that are specifically capable of activating naive T cells. Other cells which participate in antigen presentation are not actually capable of activating the naive T cells and therefore are not called antigen presenting cells. So let me just get rid of that. Antigen presentation is what I meant to write there. Okay, so I'm now going to discuss the process of antigen presentation that is occurring in all nucleated cells all the time. So this is a normal cell in the human body that is not currently infected with the intracellular pathogen. We'll come on to what happens when it's infected with the intracellular pathogen, and we'll come back to answering our question in just a moment. Okay, so what happens then is these cells all the time, they produce MHC class 1 proteins of the 3 different types that we've talked about. So HLA-A, HLA-B, HLA-C, they'll produce all of those. They'll do the same function. So we'll just refer to them all as MHC class 1. So here I'll colour in my MHC class 1 protein complex here in red. Okay, so this is a MHC class 1. So they all the time produce MHC class 1, and they're going to put peptide fragments between 8 and 10 amino acids in the peptide binding groove of the MHC class 1. And where are they going to get these peptide fragments from? Well, continuously there is a process of breaking down proteins. So I've told you that cells all the time have to get rid of proteins that have expired effectively, proteins that they've had for a long time that they've got good use out of, but which are starting to be damaged and need to be replaced with a fresh new protein to do the job better. And I've talked about one way in which cells break down proteins that they want to get rid of, which is that they send them to the lysosomes to be broken down. We discussed that when we went over phagocytosis, but there is another way, which is via the proteasome. That's what the proteasome's major function inside cells is, to act along with the lysosome to break down old proteins. Okay, so a lot of old proteins that the cell no longer has any use for and which have been damaged potentially in some way are now going to be broken down into little fragments by the proteasome. So here I'll just colour in the proteasome. So again, I'll remind you, the proteasome is just a tube, and proteins are inserted in through this tube. They go in one end, and fragments of proteins come out the other end. So on the internal aspect of the tube are loads of protease enzymes, which will just cut the protein up, the polypeptide up, into fragments. It doesn't quite cut it down into individual amino acids, but it cuts it into small fragments. So loads of the cell's own proteins. So let's just say this is a protein of the cell here, and I'll just colour that in green. Loads of proteins of that the cell is producing are going to go through the proteasome here. I'll just label the proteasome up, so proteasome, and they're going to be broken down into little fragments, and of course these little fragments can then be loaded on the MHC class 1 protein complexes, and we know the story of how they end up being loaded on the MHC class 1 protein complexes. They're going to be transported into the rough endoplasmic reticulum by the transporter associated with antigen presentation here, which I'll colour in blue. So in come these fragments into the rough endoplasmic reticulum via the transporter associated with antigen presentation, and then they'll be loaded onto the MHC class 1 protein complexes, like so. And then these MHC class 1 protein complexes, through the classical secretory pathway, i.e. via the Golgi, then into a secretory vesicle, will end up being placed on the cell membrane. So I'll just draw this here. So here now is an MHC class 1 protein complex with a self-peptide fragment presented in its peptide binding groove there in green. Okay, so this process is occurring all the time. So most nucleated cells in your body are covered in MHC class 1 protein complexes, and in their peptide binding groove they will have self-peptides. Now this doesn't normally do anything, and the reason it doesn't normally do anything is that you should not be producing effector cytotoxic T cells that are targeted against MHC class 1 protein complexes with self-peptide fragments in them. So this shouldn't normally cause a problem at all. Of course, in autoimmune responses it can go very wrong, but normally it shouldn't cause any problem at all. And the idea of this, what's the point of this, you might now ask? Well, the point of this is that it lets the cells of the immune system know about proteins that are inside this cell. It gives them a sense of what proteins are present inside the cell. So it's a part of the immune system. All cells in your body effectively are having to show what proteins they are actually producing inside of them. It's a display of what proteins are actually inside of me. Now, why is that something we'd want? Well, if you can imagine now what will happen if this cell does become infected by an intracellular pathogen. So let's imagine now here is an intracellular pathogen. Well, the intracellular pathogen will produce all sorts of proteins of its own, okay? The antigens now. So here is the intracellular pathogen, coloured in here in red, and here is, for instance, an antigen. So the intracellular pathogen will now fill the cell with lots of proteins that are the pathogen's proteins. And now what will happen is this exact same process will occur. This process is occurring all the time. It's taking samples of proteins all the time from inside the cytoplasm of the cell, and it's presenting fragments of those proteins on its surface attached to MHC class 1. So now what's going to happen is the antigens of this pathogen, they're all going to get chopped up, and fragments of them are going to be put on MHC class 1 protein complexes on the surface of the cell. And now this is going to be a sign to the cells of the immune system that there is an intracellular pathogen actually present within this cell. So I hope you understand what the purpose of this antigen presentation that is occurring in all nucleated cells of the body actually is. It gives the cells of the immune system an idea of what proteins are actually present inside that cell, and therefore it allows them to judge whether there is an intracellular pathogen inside that cell. I want to repeat that you do not refer to all cells of the body as antigen-presenting cells. Antigen-presenting cells are the special cells which are capable of activating the adaptive immune response. Most cells aren't, even though they do have MHC class 1 on their surface, they cannot activate the cells of the adaptive immune response. This cell is not going to migrate off to lymph nodes to go and meet T cells. It also isn't going to produce co-stimulatory molecules, so it is not going to activate T cells. But the role of this instead is to give the cells of the immune system, the T cells, in particular the cytotoxic T cells, because they're the ones that actually participate in cell-mediated immune responses, i.e. looking inside of cells and deciding whether the cell needs to die. It's going to give them insight into what proteins are actually present inside the cell, and therefore whether the cell is actually infected by a pathogen. Okay, so let's say that this cell is actually infected by a pathogen, and and therefore it's going to have the antigen which we've produced these effector T cells, cytotoxic T cells against, present inside of it. So this antigen is now going to get chopped up by the proteasome into lots of little fragments, and occasionally it will get chopped up in the correct way that the specific fragment that these cytotoxic T cells are directed against will actually end up being presented on the surface of this cell in the context of MHC class 1. So what overall is going to happen here then, this infected host cell is going to put on its surface the antigen fragment that these effector cytotoxic T cells are directed against attached to MHC class 1. And that is the way that these effector cytotoxic T cells are going to recognize that this cell is infected with the pathogen that they are tailored against. So, what's going to now happen is the effector cytotoxic T cell is going to come into the affected tissue, and it's going to go around looking for cells that have on their surface MHC class 1 with the peptide fragment that it is tailored against, i.e. that its T cell receptor can bind to if it's in MHC class 1 protein complex. And what's now going to form is one of these little complexes where the T cell receptor and the CD8 will bind to the MHC class 1 with that peptide fragment on its surface. So I know you're probably fed up of seeing these pictures, but I am going to draw it one more time here. So this is going to be the infected host cell, so this is the surface of the host cell here. And then here, this is the MHC class 1. And here now, this is the peptide fragment of the antigen from the pathogen, and it's the specific peptide fragment that this effector cytotoxic T cell is targeted against. So remember, the effector cytotoxic T cells, they have a T cell receptor that is directed against not a specific antigen, but more specific than that, a specific antigen fragment, so a specific fragment of that antigen. So it's more specific than just being directed against the antigen. But if this cell is infected by this pathogen, then because it's doing this process so much, it will eventually happen to cut up the antigen in the correct way to get the correct fragment presented on at least one of its MHC class 1 protein complexes, so that it can now be recognized by this effector cytotoxic T cell. You also have to remember that we don't just initiate one adaptive immune response, we initiate loads of adaptive immune responses. So you won't just be producing one type of effector cytotoxic T cell, you'll be producing effector cytotoxic T cells that are targeted against all sorts of different antigen fragments from many of the different antigens of this pathogen, and they'll all be coming into this area and trying to find MHC class 1 protein complexes with their specific antigen fragment presented in the peptide binding groove. So it's likely that one of them will find the infected host cell. Okay, so there's the host cell membrane and the MHC class 1, and I was just about to colour it in. So here it is in red, like so. And here now is the peptide fragment. And now what's going to happen is the T cell receptor, it will be specific to this MHC class 1 with this specific peptide fragment in, and oh joy, look, it's managed to find it. It will now bind to it. So here This is the T cell receptor of our effector cytotoxic T cell. So I'll just label this up. So this is the T cell receptor here, and this cell surface here is our effector cytotoxic T cell, so I'll put TC cell. And now, of course, what's the final thing that you know is going to bind here? Well, the effector cytotoxic T cell, its co-receptor is going to be CD8, which will bind to the MHC class 1, so I'll just draw that in here. So there is the CD8 molecule, and I'll colour that in orange. Okay, right, so what was I about to label this up as? Okay, MHC class 1, I think. Right, so this complex is going to form, and now once this complex has formed, this is the answer. This is how the effector cytotoxic T cell knows that this host cell is infected with the pathogen. It has found on the surface of this host cell an MHC class 1 molecule with the specific peptide fragment that it is tailored against. Its T cell receptor is perfect for binding to this MHC class 1 complex with this peptide fragment in. Okay, it is an effector cytotoxic T cell. It's now going to say this cell needs to die. It's infected with a pathogen, it needs to die. So it's going to induce apoptosis in the host cell. And the question I now want to answer as the final part of the cell-mediated adaptive immune response is, how does the effector cytotoxic T cell get this host cell to die? So it induces apoptosis in the infected host cell, and it does so by two different mechanisms. So let me go over these two different mechanisms. So I'll start off with mechanism 1. So mechanism 1 involves the Fas ligand and the Fas receptor. So what's going to happen is our effector cytotoxic T cell is now going to activate the Fas receptors on the surface of the host cell. So this now is going to be the membrane of the host cell, and this is going to be the membrane of the effector cytotoxic T cell. So if this recognition has occurred, if this complex has occurred, then what's now going to happen is the effector cytotoxic T cell is going to activate receptors on the surface of the host cell known as Fas receptors. Now, all cells in your body will have Fas receptors on their surface, or at least most cells in the body will have Fas receptors on their surface. And these are receptors for the activation of apoptosis. So if you activate Fas receptors on a cell, it starts to activate the apoptosis process inside the cell. So this is a pathway known as the extrinsic apoptosis pathway. And I'm not going to go through this pathway in detail. I do have a video somewhere on my channel which covers the extrinsic apoptosis pathway. I think it might be in the playlist on cancer. So it's going to activate something known as the extrinsic apoptosis pathway, which is going to lead to the cell committing apoptosis. Okay, and the molecule that the effector cytotoxic T cells use to activate the Fas receptors is known as the Fas ligand. So they're going to activate the Fas receptors on the surface of the host cell that's infected with the Fas ligand. So here in yellow, this is the Fas ligand. Right, so the Fas ligand on the surface of the effector cytotoxic T cell will activate the Fas receptors on the surface of the host cells. This will result in a pathway known as the extrinsic apoptosis pathway occurring inside the host cell, and overall what this results in is the activation of procaspases to caspases. So remember, in order for apoptosis to occur, what you have to do is convert inactive precursor enzymes inside all cells, which are known as procaspases. Okay, so this rectangle here is denoting the procaspase. This is an inactive precursor to a caspase enzyme. So there are loads of enzymes that are in an inactive state inside the cytoplasm known as procaspases, and they're doing nothing. They're waiting to be activated, and if they are activated, then they're now known as caspases, and once they're in this caspase state, they go around and break down everything. So it's effectively like a whole bunch of enzymes inside the lysosome sitting in an inactive state waiting to be activated in the cytoplasm of cells. They are lytic enzymes. They break down biomolecules, and if you activate these enzymes, they're going to break down everything inside the cell, and that's the way in which the cell commits apoptosis. So all cells effectively have these bombs waiting to go off to kill them, known as procaspases, and once they've been activated they become caspases. And you can see I've shown now the caspase as actually having an active site, and therefore it's actually going to be active and it's going to break things down. Okay, so the extrinsic apoptosis pathway, overall what that results in is the conversion of procaspases into caspases. The caspases will then break down everything inside the cell, and that's how the cell commits apoptosis. So once you've activated caspases, that's when the cell is devoted into the apoptosis pathway. There's no escape once you've got enough caspases activated, you're just going to be destroyed. Okay, so that's one way in which the cytotoxic T cells are going to kill the host cell that they have decided is infected. There is also a second pathway, which I want to add on here. Okay, so the second pathway is that they secrete vesicles onto the surface of the host cell that contain granzyme enzymes and also perforin proteins. So let me just show this here. So inside the effector cytotoxic T cells, you have these special vesicles which contain an enzyme known as granzyme, so they contain loads of these molecules, not just one. So they'll contain granzymes, and they'll also contain proteins known as perforins. Okay, and what's going to happen is once the effector cytotoxic T cell has decided that the host cell that it's sitting next to, bound to by this interaction here, is actually infected, it's going to fuse these vesicles with its plasma membrane and therefore release the horrors within, the granzymes and the perforin proteins, onto the surface of the host cell, the infected host cell. So here, this is supposed to show the vesicle now fusing with the plasma membrane, and therefore the granzyme and perforin will be released onto the surface of the host cell here. Okay, so let's now actually discuss what granzyme and perforin are going to do. So these two work together, they are accomplices. So perforin is going to form pores on the surface of the host cell membrane. So we've seen lots of proteins that are capable of actually forming pores on the surface of cell membranes. Defensins and complement proteins, the membrane attack complex and complement. Now we're seeing another one, perforin. So perforin proteins are capable of joining together to form a pore on the surface of the cell membrane, like so. Okay, and I'll just colour this in blue. So, as always, this is going to promote osmotic lysis of the cell, but there's a more important thing that perforin is actually going to allow through into the inside of the cell in this case, and that's the granzyme enzymes that have also been released onto the surface of the host cell with the perforin proteins. So the perforin proteins, they form a pore on the surface of the host cell, and what that now allows to enter into the cytoplasm of the cell is the granzyme enzymes. So here, this now is the granzyme enzyme, so I'll colour it in green. So here's the granzyme enzyme. And now what do the granzyme enzymes actually do once they're inside the cell? Well, they're going to activate the conversion of procaspases into caspases. So they act on the procaspases to activate them into caspases, and therefore they kickstart the apoptosis pathway. So perforin and granzyme, they work together in order to make the cell commit apoptosis. The perforin forms a pore that then allows the granzyme to enter into the cytoplasm of the cell, where it's going to activate procaspases to caspases and therefore trigger apoptosis of the infected host cell. So, that now is the cell-mediated adaptive immune response completed. We have been through antigen presentation of these antigen fragments from the intracellular pathogen. We've been through how you will activate CD8 positive naive mature T cells. We've been through how the mature naive T cells will clonally expand once they've been activated, and then they'll differentiate into memory cytotoxic T cells and also effector cytotoxic T cells. We've then seen how the effector cytotoxic T cells will go into the bloodstream and they'll come to the affected area, into the tissue fluid of the affected area, and what their job is to do is to kill if infected host cells, and of course when the infected host cell kills itself, the pathogen that's within it will die as well. Of course, that's the whole point of this exercise. So how then does this actually occur? We've discussed how all nucleated cells within the body, well, pretty much all nucleated cells within the body, are participating in a continual process of antigen presentation, where they chop up proteins from within their cytoplasm and put fragments of those proteins on MHC class I proteins on their surface. And the function of this is that they are continually displaying what's inside me. These are the proteins inside me. These are fragments of the proteins inside me. And usually, when they're just putting fragments of proteins from their own proteins on their surface, that's absolutely fine. No cytotoxic T cells will hopefully be being produced that will be capable of recognizing those MHC class I peptide fragment complexes. So the cell won't be in trouble from any effector cytotoxic T cells. However, if you do get infected by a pathogen, then you'll start actually getting fragments from the pathogen's proteins presented on your surface on MHC class I proteins, and now these can be recognized by effector cytotoxic T cells, and they then know that this cell is infected, and therefore they unleash apoptosis signaling on it. And there are two ways in which they get the cell to commit apoptosis. One involves this fast signaling process, where they activate the fast receptors on the surface of the infected host cell using the fast ligand, and the other involves releasing their vesicles, which contain the granzyme and perforin proteins. The perforin proteins form a pore in the cell membrane, and the granzyme enzyme goes inside to activate the apoptosis machinery, and therefore the cell will commit apoptosis, and that will cause the annihilation of the pathogen that was inside that cell. Okay, so that is the cell-mediated adaptive immune response complete. That is the adaptive immune response to intracellular pathogens. So if you've got a pathogen that is intracellular, it will activate a cell-mediated adaptive immune responses. And let me stress that although we have followed this just for one antigen fragment of one antigen, each antigen will have loads of antigen fragments, and each pathogen will have loads of antigens, and this same thing can be happening for all of those different antigen fragments that you can possibly come up with. And therefore you'll get loads of these cell-mediated adaptive immune responses all occurring in parallel. Okay, and the idea then is that we can clear the pathogen that is inside of host cells in this way. In the next video, we will go on to the humoral adaptive immune response, which is the portion of the adaptive immune response initiated to extracellular pathogens. And remember that often an intracellular pathogen will also be extracellular at some point whilst it's going between host cells, and therefore whilst it's in the extracellular portion, it will often have humoral adaptive immune responses initiated against it. So not only do solely extracellular pathogens get humoral adaptive immune responses initiated against them, but also intracellular pathogens that have to go extracellularly to go between cells, they will also get humoral adaptive immune responses initiated against them. Okay, so we'll do that in the next video.